Hi, my name is Vuk Đorđević. I'm a master's student at the School of Electrical Engineering in Belgrade, and this is my project for genome informatics. Single-cell analysis of how human cells respond to nanoplastic particles of different sizes, where nanoplastics are tiny particles now found in human blood cells in direct contact with our immune cells. My question is whether size matters. Do small and large nanoplastics cause different responses, and does a mixture of both do something neither size does alone? To answer it, I use single-cell RNA sequencing, which reads which genes are active in each individual cell. The data is 4 samples from a single donor, peripheral blood immune cells exposed to 40 nanometer particles to 200 nanometer particles, to a mixture of both, and 1 unexposed control. About 34,000 cells in total. One thing to be upfront about, there is one donor, one sample per condition, so no biological replicates. That shaped how I did the statistics, and I flag it throughout. The whole analysis runs through one pipeline of 6 stages: quality control, integration, annotation, composition analysis, and differential expression and site-specific effects. A single command reproduces all of it from the raw data, and each stage has automated tests. The first stage is quality control, removing low-quality cells before trusting anything. These 3 plots are via lens, basically histograms on their side. Fatter where more cells sit, along the bottom are my 4 samples. Left is how many genes each cell expressed, middle is total reads per cell, right is the percent of mitochondrial reads, a sign of dying cells. This is after clearing, and the shapes look healthy and similar across all samples. I set the cutoffs from the real distribution, not by guessing. Drop cells with too many genes, usually 2 stuck together, and cells with too much mitochondria, which are dying. This is the same cleaning step, drawn in a different way. Every dot here is 1 cell. Left to right is how many reads the cell has, and bottom to top is how mitochondrial it is. The red dashed line is my cutoff at 15%. Almost all the cells form a dense cloud hugging the bottom with low mitochondria, which is exactly what healthy cells look like. The handful of dots floating up above the red line are dying or broken cells, and these get thrown out. The second stage is integration. Both pictures are a UMAP. Each cell has thousands of genes, and UMAP squashes that down to 1 dot on a map, so cells with similar gene activity land close together. The axes are just map coordinates, only closeness matters. Each dot is a cell colored by sample. I want the colors well mixed. If cells clumped by color, the sample would matter more than the biology, which is a technical artifact. Here they're nicely blended, so cells group by cell type, not batch. One honest caveat is with a single sample per condition, batch and treatment are tied together. So for the gene level results later, I use the uncorrected data where this can't bias anything. The third stage is labeling the cell types. On the left, the same cell map colored by cell type, which is T cells are the big purple mass on the right. Monocytes are the orange island on the top left, B cells blue at the bottom, NK cells are green. Clean separate islands mean the types are real. On the right, a dot plot that proves the labels. Rows are clusters, columns are marker genes like CD3D for T cells or CD14 for monocytes. A big dark dot means that gene is strongly on in that cluster, and the dark dots line up on diagonal, so each cluster lights up its own marker. I labeled with a tool called Cell Typist and cross-checked against 2 independent methods, and they agreed at about 93%. The fourth stage asks whether exposure changed how many of each cell type there are. The bars show what fraction of a sample is each cell type, and the 4 bars per group are the 4 samples. T cells dominate every sample. The tall bars on the right at around 80%, which you can see on the far right here. I covered the T cells letters. The thing to notice is the monocyte group. The green 200 nanometer bar is noticeably taller, roughly 2.5 times the control's monocyte fraction, while everything else barely moves. So 200 nanometer specifically bumps up the monocytes. With no replicates, I report this as a descriptive shift and treat any test as exploratory. The fifth stage asks which genes actually change on exposure within each cell type. In both bar charts, bar height is the number of genes that significantly changed. The left chart groups by condition, colored by cell type. The orange monocyte bars tower over everything because monocytes eat and clear foreign particles, which is plastic in this case. And the 200 nanometer bar beats the 40 nanometer one, so bigger particles, bigger response. The right chart is the sixth stage, showing where those genes overlap. 4 bars per cell type, genes reacting only to 40, only to 200, to both, and the key one in red, genes that fire only in the mixture and in neither size alone. In monocytes, that red bar is about 180 genes, a whole response that only appears when both sizes are combined. So let me pull the main findings together. First, monocytes show the strongest response to nanoplastic of any cell type. Second, the 200 nanometer particles drive more cell type unique genes than the 40 nanometer ones. And third, the biggest one, the 40 plus 200 nanometer mixture produces an emergent monocyte response, about 180 genes that are significant only in the mixture and in neither single size. Interestingly, in the lymphocytes, the opposite happens. The mixture is actually weaker than either size alone. Either way, the combination of the two sizes does something the individual size does not. So what's the new thing? I ran pathway enrichment on those 180 mixture-only monocyte genes, and it points clearly at the inflammation. The top pathways with strong statistical support are interleukin, cytokine, and TNF signaling. And the last one is the program a monocyte runs during a bacterial infection, except here it's set off by plastic. And two of the strongest genes, the RIP-K2 and TRA-F1, coordinate immune and TNF genes. So for 40 and 200 nanometers each cause small subthreshold changes, but together they push the monocyte over the edge into an inflammatory state. And since real exposure is always a mix of sizes, testing single sizes alone could underestimate the risk. For the additional insights, part I implemented 5 extra analyses, which this slide lists, and the next slide shows each one. They are module scoring, a mixture additivity check, a clustering robustness check, a ligand receptor communication analysis, and dose response. So the first additional analysis is module scoring, a color grid where each row is a sample and each column is a biological program, a named group of genes, oxidative stress, inflammation, interferon, and heat shock. Red means the program is turned up, so the whole oxidative stress column goes deep red in all 3 exposed samples, but not the control. So plastic reliably stresses the cells, and the inflammation column turns pink specifically under 200 nanometers. So the size effect shows up here too in independent gene programs. The second analysis is a direct test of whether the mixture is just 40 plus 200 added together. 1 scatter per cell type, each dot a gene. Left to right is what I predict if the mixture were the two sizes summed up. Bottom to top is what it actually did. And the red diagonal is where the prediction would be perfect. So if every gene set on that line, the mixture would be boringly additive, but in the monocyte panel there are clear we can see clear blobs of genes off the line. Genes in the mixture switched off or on that you never predict from the single sizes. So that off the line behavior is exactly the emergent non-additive effect. The third and fourth analysis is the left plot is a sanity check on my clustering. So the green dashed line is how much the grouping still agrees with my default setting, where 1 is identical, so it stays high, so my cells will run into fluke of 1 setting. The right plot is a cell-to-cell messaging. Each row is a signaling pair, and the bar goes right if that message got louder after exposure. So the standout is IL-1B, a classic inflammation alarm shooting far right in all exposed samples and furthest under 200 nanometers, so the cells are shouting inflammation at each other. The fifth analysis close response I just showed on the earlier D slide. And finally, to be clear about the limits, 1 donor, no replicates means the differential expression is done per cell and is exploratory with a pseudo-replication caveat. There is no external gene level reference, so I validated the results internally and biologically, but everything reproduces from the repository with a single command of full test suite, so that's it. Thanks for watching.